this video we'll be looking at the topic of reproduction. Now this is actually quite a large topic, even if you're just doing combined biology. So there's a bit about cell division in terms of meiosis in particular, comparing it to mitosis, which you've learned earlier on in the course. But then the whole chapter is really focusing on patterns of inheritance. So looking at how we can have different types of genes, and looking at the combination of those genes, we can result in lots of different physical characteristics that are being shown. So we're going to be spending this video looking at that aspect, and then later on in the video there will be some separate only content when we go into details about protein synthesis, which is about how our DNA actually codes for the structure of proteins. So first of all, let's do a quick overview on the concept of sexual reproduction. Now, in sexual reproduction, it's always about using two particular parents, so a male and a female, and both of them are able to produce gametes. So for a male, they will be able to produce sperm cells, for example, and for the females would produce egg cells, like this. Now, the interesting thing is that for the individual parents, they have what we call diploid cells. So or DI, think of double, or we can sometimes use the shorthand 2N for this one. Diploid cells means that they have the full set of chromosomes. Now, what needs to happen is that the diploid cells need to split twice to produce haploid cells, which are the gametes. So think of haploid, like half, so they contain half the number of chromosomes compared to diploid cells. And this process requires meiosis to occur, which is the cell division that produces haploid cells. So now let's have a quick look at the process of meiosis. Now, in meiosis, the idea is that the cell's going to split twice to produce half the number of chromosomes, and you will end up with 4 daughter cells. So it's quite different to mitosis, which is what you've learned in the cell division chapter, and we're going to look at the differences shortly. But let's start with meiosis first. So let's say, for example, that the cell in the top here, let's say this is a diploid cell, and in this picture, in this example, I'm showing 2 chromosomes only. Now, to start with, in the beginning, what happens first of all is that the chromosomes can duplicate themselves, which turn them to something that looks like this. So each of the chromosomes has duplicated, and they're attached in the middle. Now, after that, what was interesting is that actually the chromosomes would line up along the center of the cell, and they would separate into 2. So it's actually going to separate a bit like this. So you get one chromosome on the side and the other chromosome on the other side. Now, after that, each of those chromosomes would separate, or shall I say that each of the arms of the chromosomes is going to separate again, and then we end up with 4 cells, each containing one half of the one set of the chromosome compared to before. So if we look at the beginning, we started with a diploid cell, which has 2 chromosomes. We ended up with 4 cells, each containing 1 chromosome. So it's half the number of chromosomes, therefore we call it haploid. So again, if we compare to mitosis, is that both of them requires the DNA to duplicate to start with, but then the difference is in meiosis, the cell splits twice to form 4 genetically different daughter cells, whereas in mitosis, the cell splits once to produce 2 genetically identical daughter cells. So we can summarize it in a table like this. Now, there are a few more differences. I mentioned it briefly already. In mitosis, the way that the chromosomes actually separate is that one set of each chromosome separate after duplication. So the daughter cells are clones, or what we mean by, or we can say that they're genetically identical, right? So the DNA, it's the same. But for meiosis, what happens is, in GCSE, you don't need to know this in detail, but if you do A-level biology, we go into detail about exactly how the DNA interacts in meiosis, but there will be a bit of mixing of genes. So this one, we say that there is going to be some genetic variation in the daughter cells. So the daughter cells are all completely different from each other, whereas in mitosis, they're exactly the same. Also, as mentioned, that partly due to that, so we say that in mitosis, you result in the daughter cells will have the same number of chromosomes compared to the parent cell, whereas in meiosis, you would have half the number of chromosomes. So in an exam question, if they ask you to compare mitosis and meiosis, you could say that, or you can simply say that they produce the diploid cell would undergo mitosis to produce two diploid cells, but whereas in meiosis, one diploid cell can be divided into making haploid cells. So those are the four differences between mitosis and meiosis. This could easily be a 3-mark question, so make sure that when you're phrasing your answers, indicate exactly which cell division you're referring to. Don't say that one makes 2 cells and the other makes 4 cells. That will not get you any marks. You want to say mitosis makes 2 cells, but meiosis makes 4 cells. So that is about meiosis. Let's go back to the process of sexual reproduction. So after the parents individually gone through meiosis to produce the gametes, the sex cells, so like they said, in the males it will be sperm cells and the females would be egg cells. So for the case of humans, it would be 46 chromosomes starting from the human cells, ending up with 23 chromosomes in the gametes. Now after that, what happens is that the gametes can fuse together, and this process is called fertilization. And after that, you make one cell like this, but because you're fusing the gametes, which both carry 23 chromosomes, you ended up back into a diploid cell with 46 chromosomes again. So we said that one of the importance of meiosis is that you creating haploid cells, so therefore that you can, when they go through fertilization, they restore the original number of chromosomes, like in this case 46. Now this resulting fertilized egg is called a zygote, right? Which, as I mentioned, is a diploid cell. So it's simply a fertilized egg. Now, however, we are not made up of one cell, we are made up of multiple cells. So what happens is that this cell has to go through mitosis to replicate itself and just to make more. So this is mitosis, where they make genetically identical daughter cells, and this ball of cell is now called an embryo, which we hear quite often when it comes to, for example, say in IVF, we will be implanting the embryo into the mother. And after that, the embryo stem cells basically can continue to divide and make more of themselves, but they can also go through differentiation. So a quick recap is that the embryonic stem cells are undifferentiated cells, unspecialized, and they have the ability to differentiate or specialize into all types of cells. So some of them become nerve cells, some become muscle cells, some become red blood cells, etc. So after some time, we can get something like this, which is a baby, but we use the word fetus to describe or to refer to the baby. Okay, so the fetus is the developing baby inside the mother's womb. And essentially, the fetus will continue to grow, do more mitosis and differentiation until they become a full-grown baby, and then the mother gives birth, the baby grows up into a whole adult human being, then the cycle continues in sexual reproduction. So how is sexual reproduction different from asexual reproduction then? So keeping in mind that asexual reproduction is basically cloning. You're making genetically identical organism from the parent. So let's draw a table to compare the two. So I'm keeping the diagram on the left-hand side to just for reference as we go through this list. So first of all, number of parents. So in asexual reproduction, you only need one parent. Okay, so only one organism is needed, whereas in sexual reproduction, you need 2 parents, right? Because it involves the mixing of genes. So on that note, sexual reproduction requires fertilization to occur. So we know how the gametes can fuse together to mix the genes, whereas in asexual reproduction, there is no fertilization. You're literally just cloning yourself. As I mentioned, you produce clones or genetically identical offspring, whereas sexual reproduction, you got genetic variation or genetically different offsprings. In terms of the process itself, asexual reproduction only requires mitosis to occur, because again, it's the process where you make genetically identical cells, whereas in sexual reproduction, you need both. And this is a common misconception. A lot of students tend to think that asexual needs mitosis only, sexual means it only needs meiosis. But in fact, sexual reproduction needs both, because meiosis only produces the gametes, but then that there are just still individual cells. You need to make sure the gametes fuse together to grow into a zygote, which then goes from mitosis to grow into whole offspring. So sexual reproduction requires both mitosis and meiosis, not just one. Other aspects to consider as well is that asexual reproduction occurs much faster because you only need one parent. So, and on that note, it's more energy efficient. So it requires less energy because the organism doesn't need to find a mate to do this, and it just needs to clone itself. There's no fertilization involved, which means less energy is needed. Whereas in sexual reproduction, is the opposite. It is a slower process and also requires more energy. But the benefit is that you produce genetically different offspring, which means again increasing the chances of the organism surviving if the environment changes. So again, this is a typical question in an exam, so it could be phrased as a 3-mark question where they ask you to describe 3 differences between the 2 types of reproduction, or it could be a table where they would ask you to complete that table to compare them, or it could be a tick box question. So they would have some bits on the side. For example, mitosis is involved. Then you can take that you need mitosis in both asexual and asexual reproduction. So it could be a tick box question. It could be a slightly more wordy question where you have to describe the 3 differences. Another type of question will be evaluating the 2, especially if you're doing separate biology. Then they might say, describe the benefits or the disadvantages of these 2 types of reproduction. Then again, you look at the fact that, okay, asexual reproduction is faster, less energy, only needs one parent, but then you are creating genetically identical cells, so less variation, higher chance of dying, basically, if the environment changes. And then it's essentially the exact opposite in the sexual reproduction bit. So that section just now summarized the differences between asexual and sexual reproduction, and we looked at mitosis and meiosis as well, briefly looking at the comparison. As I mentioned, these are very typical exam questions, so it's important that you understand them. So now we're going to look at the patterns of inheritance. Okay, so let's come back to this picture here to kind of hook our new learning bits onto this diagram. Okay, so we're going to focus a bit more on the fertilization bit, which is when the gametes fuse together, which is the mixing of genes. Now, what we say in that process is that the gametes will be carrying different types of genes. So, for example, both the mom and the dad would be carrying a gene for eye color, but depending on the parents, sometimes the parent might have blue eyes, sometimes they might have brown eyes, for example. That means even though they have the same gene, the gene is slightly different from each other, allowing the different combinations of that to produce different characteristics. Those different types of genes are called alleles. Now, it's important that in exam questions, when you're asked to define alleles, which can happen, you want to say it's different versions of the same gene, right? So you can't say that there are different versions of genes as a general term, because yes, we do have many, many different genes, but they all code for different things. But if you say it's different versions of the same gene, it means that the gene is coding for one particular protein or one particular characteristic, but there will be a slight variety in that particular characteristic. So we're going to take a pause there and actually look at the structure of how the DNA might be organized here. So first of all, we got the term DNA. Now, DNA is referring to the actual molecule, biochemically speaking, the molecule that makes up all of the chromosomes, right? So we say that the DNA molecule has this particular structure. So it's made up of 2 strands and they're twisted together. So we got the term double helix. Because it's made of 2 strands, therefore double, and is twisted together, helix. So the DNA is the molecule that carries the genetic information based on their base sequence. So if you're doing separate biology, I'll talk a bit more about that later, but the idea is that there is a very particular sequence in the DNA that codes for the actual structure. Now, the DNA is the actual molecule that make up all of these structures. So we say that a bunch of molecules, a bunch of DNA becomes a gene. So for example, if we're looking at the double helix here, so the gene would be referring to a section of it. Now, I understand that this one might be a bit confusing because we said that DNA is the simplest molecule that makes up the gene. So why does it look like as if the gene is smaller than it? It's because the DNA is the fundamental ingredient, shall we say, that makes up everything. But if the whole structure itself has a double helix structure, so it could be as small or as big considering how we look at this. But essentially, we say the DNA is the molecule that makes up all of the genetic information, and then we say a gene is a section of DNA that codes for a protein. And finally, when we got lots of genes together on a particular structure, we call that a chromosome, which is found in our nucleus, as we know before. So a chromosome would essentially, we typically draw it like this, okay, and we find them inside the nucleus. So it's a structure that carries the genes and is made up of DNA found in the nucleus. So what we are saying here is that essentially the allele is when we have one gene, but we can have different versions of it. Most genes could have two alleles. So an allele can be dominant or it can be recessive. So dominant allele, we usually represent that with a capital letter. So for example, I would use the letter B, then it will be represented by the big letter B, whereas recessive alleles is represented by a small letter, so for example in this case small letter B. So as the name kind of implies, the dominant allele basically overshadows the recessive allele. So a dominant allele is basically saying that you only need one dominant allele for its characteristic to be shown, whereas for a recessive allele you need to inherit two recessive alleles or two copies of recessive alleles in order for the recessive characteristic to be shown. So we say that in fertilization, what is happening is that the egg cell would be carrying one specific allele of that gene. So for example, the egg cell might be carrying the dominant allele, whereas the sperm cell might be carrying the recessive allele instead. And when they fertilize, the zygote would have received and inherited one dominant and one recessive allele. Or it could be that both the sperm cell and the egg cell have dominant alleles. That means the zygote would be inheriting two dominant alleles. Or if the sperm cell and egg cell are both carrying a recessive allele, then the zygote will be inheriting two recessive alleles. And so therefore you can have three possible combinations here. As I mentioned earlier, you could be inheriting two dominant alleles, and in that case, we call this homozygous dominant. So homo meaning the same, zygous like a zygote. So therefore, homozygous dominant means you're receiving two of the same dominant allele. Similarly, if you're inheriting two recessive alleles, then it is called homozygous recessive. Now, obviously, the combination is if you receive one dominant and one recessive allele, then we got a slightly different term. So we mentioned that homo prefix means the same, so different means hetero. So heterozygous refers to when you inherit one dominant and one recessive allele. So if we were going to use the letters to represent this, then homozygous dominant would be big B, big B. Heterozygous would be big B, small b, and homozygous recessive would be small b, small b. These three combinations of alleles, we have a term to refer to this. We call this their genotype. So I guess you can say the type of gene, but essentially it means the allele combination. Now the genotype, the allele combination, essentially determines your phenotype. So phenotype, think of physical. So this is referring to the physical characteristic or the physical trait that is going to be expressed by the person or by the offspring that inherits this. So in this case, for example, both the homozygous dominant and heterozygous, they would show the same trait, which is they would both show the dominant trait. Because as I mentioned earlier, the dominant allele, you only need one of the dominant allele for the dominant trait to be shown. So therefore, even if you're heterozygous, the dominant allele would basically overshadow the recessive allele, and it would show the dominant trait. Whereas for the homozygous recessive one, because they're both recessive, therefore it would show the recessive trait, if that's the genotype. So when it comes to inheritance, sometimes we can use something called a Punnett square to basically kind of predict or try to explain some of the inheritance patterns. So for example, if I know what the parents' genotypes are, I could predict what genotype their baby might have, or what is the probability of the different genotypes and therefore the phenotypes. So we can have many different combinations here. So let's start with, let's say, a homozygous dominant person crossing with a homozygous recessive person. So we say that in this case, the homozygous dominant person can only make gametes that contain the dominant allele. So let's say if I use a circle to represent the gametes, then this is what they would have. Whereas the homozygous recessive person can only make gametes carrying the recessive allele. So when it comes to fertilization, it could be any one of the gametes made by these two people fusing together. So we can say that it's a random fertilization. So if the first first gamete here fuses with this gamete, then it would produce a zygote that is heterozygous like this. And then same with the other one, if we use the Punnett square and show how the two random gametes might be able to produce these zygotes. So remember that we're not saying they're going to make 4 zygotes and 4 babies all the time. We're just saying that it could be one of these 4 possible combinations. So in this case, you can see we say that the zygote is 100% heterozygous. So therefore, we mean that there is going to be 100% a chance for the baby to show the dominant trait. So I can use that to predict what traits the baby might have. So let's look at a slightly different example. Let's say this time I've got 2 heterozygous people, a couple who are both heterozygous like this. That means both of them, let's say the dad, will be able to produce sperm cell that carries the dominant allele and also sperm cell that carries the recessive allele. It's possible. When during meiosis they are going to be separated. Same for the mother. The mother can produce egg cells that contain the dominant allele, but also can make egg cells that carry the recessive allele like this. So when, like I said, random fertilization occurs, any one of those sperm cells can fuse with any one of those egg cells. So this would then become our predicted outcome. So you can see this time it's very, very different. We say that there is a 25% chance or 1 in 4 chance for the baby to be homozygous dominant, whereas I have a 50% chance for the baby to be heterozygous, and again 25% chance for the baby to be homozygous recessive. So what does that mean? It means that if the couple has a baby, there is a 75% chance for the baby to show the dominant trait because of the numbers you can see here. Whereas, or you can say the baby has a 25% chance to show the recessive trait. So again, looking at the question carefully, see if the question is asking you about the chance of certain genotype, or is it the chance for a certain trait, dominant or recessive. They might give you in the question saying that this trait is caused by a dominant allele or recessive allele, and you're likely to be given a scenario where they would give you the parents' genotype/phenotype, and you have to kind of basically predict or explain what are the probabilities for the different genotypes or phenotypes. Sometimes it could be that they tell you that the baby has a 50% chance to show this trait, then you have to draw a Punnett square to prove that is correct. But essentially, you can use Punnett squares like this to showcase this one. Common mistakes in exams is that most students are able to draw the Punnett square perfectly well and interpret it well. However, exam questions want you to state very clearly which gametes are made by which parent. So for example, you might want to add a little note saying that, oh, let's say the top two, this is from the male or from the father, and then these two are from the female or from the mum. So you might want to specify and saying, okay, labeling the Punnett square like this. And if the question is asking you, let's say this trait is caused by a recessive allele, you then might want to circle this one and say, oh, this combination is going to cause the recessive condition. Okay, so typically speaking, people lose 2 marks because they didn't label the gametes, saying which parent they come from, and not indicating which genotype causes the trait. So just be careful in how you can make sure you don't lose those easy marks. Another way to use Punnett squares is to use it for gender determination or sex determination. So again, sometimes the questions might ask you to predict if the baby is going to be a boy or girl. I mean, fundamentally the answer is always going to be the same, okay? But we're going to use a Punnett square to prove that. So first of all, what you need to know is that females, at least for human females, the sex chromosomes combination will be XX. So if we were to draw this out, basically it means that there are going to be 2 X chromosomes like this. So we're not talking about alleles here, we're talking about the actual chromosome. Whereas for males, it's always going to be XY. So that means for males, they would have 1 X chromosome like this and 1 Y chromosome, which is slightly smaller. So again, we mentioned in sexual reproduction is always between a male and a female. So let's say, for example, I would have a female which produce X cells that can only carry the X chromosome, and whereas the males can produce gametes that can either contain the X chromosome or the Y chromosome. So using the Punnett square, we can see that if any one of those gametes fuse together, these are the 4 possible combinations. Right? And you can see, therefore, it means there is always going to be 50% chance for it to be a boy and 50% chance for it to be a girl. So it's always 50-50. So most of the time, if the question asks you to draw a Punnett square to either prove something or to predict something, it can be sex determination, although most of the time it's mostly about maybe looking at a inherited disorder. So there are 2 inherited disorders that you need to know. So again, we're going to be looking at this in terms of the phenotypes as well. So we got a dominant inherited disorder caused by a dominant allele, which is called polydactyly. The person with polydactyly will have an extra digit. So we mean an extra finger or an extra toes. Whereas for another type, which is a recessive condition, that will be cystic fibrosis. So cystic fibrosis is a bit more harmful. It's basically when the body produces excess mucus. So that means the mucus will clog up your airways or your digestive tract or your reproductive tract. That means it's going to be more likely for the person to have infections, and they might struggle to breathe as well. So again, questions could be that they give you a family tree diagram and ask you to use the family tree to predict whether, let's say, a couple within that family is going to have a child, and you have to predict will they have, or what is the percentage chance of the baby having one of these conditions. So a family tree diagram looks a bit like this. There is always going to be a key on the side that tells you what the symbols mean. Usually they would have a square for a male and a circle for a female, and a white bit means it's a normal or healthy person. Gray means that they have that particular condition. So for example, in this case, they might ask you to predict what is the probability of the 4th child to have a particular condition. And at this point, you kind of have to guess a little bit, because let's say if this is a recessive condition, then we know that the dad definitely has is homozygous recessive because he has the trait. And then the question is the mother homozygous dominant or heterozygous? So then you look at the other 3 offspring, you can see that, they're able to make one child or one daughter that doesn't have the condition, which means that she must have inherited 1 dominant allele from the mom and 1 recessive allele from the dad. So and you can see for the other 2, they must be homozygous recessive. That means they must have inherited 1 recessive from the dad, but the other must have come from the mom. So therefore, from that, we know that the mom has to carry the recessive allele. So therefore, we know that she cannot be homozygous dominant. So in this case, we say the mom is a carrier. She is healthy, but she carries the recessive trait. And same for this daughter, she's also a carrier. So in that case, then we can do a Punnett square again to quickly predict what genotype the baby would have. So let's say, for example, the mother would have these two gametes, whereas the dad would have these two gametes. And then very quickly, we can do a quick Punnett square, which shows this particular combination. And we know that means the baby has a 50% chance to become a carrier, a healthy carrier, but also 50% chance to show the recessive condition. Let's say if this is cystic fibrosis, then maybe the child would have a 50% chance to have a cystic fibrosis. And also, it's a 50% chance for it to be a boy or girl, because as we mentioned, the X and Y chromosomes is basically set. It's going to be 50%. So there you have it. That is a summary of the whole topic if you're doing combined biology. As I mentioned, it is a rather big topic and there's lots of new keywords that you have to learn here. But actually, most people, once you have learned what the keywords mean, you can easily pick up the marks that ask you to define them. So it'll be helpful to do do some flashcards on the definitions on one side and the keyword on the other side and just learn it. And on the other hand, most people find the Punnett squares relatively straightforward. So again, it's about making sure that you label which one's from the dad, which one's from the mom, in terms of gametes, and make sure you highlight, oh, that's going to be which characteristic is going to be the one that we're going to be looking at in that particular question, for example. And then again, linking to family trees. Again, more exam practice on the family trees would really, really be helpful. And finally, I guess the trickier ones would be asking you to give differences between mitosis and meiosis, or differences between asexual and sexual reproduction. I will be covering the separates only content in a different video. If you're doing separate sciences, then make sure you check that one out as well.